Okay, so chapter 27 is a general introduction to animal diversity and an overview of a lot of different concepts and vocabulary. Then chapter 28 and 29 will specifically delve into 9 of the phyla in the animal kingdom, the top 9 phyla. So most animals have most of these features. The only feature that all animals have is listed at the bottom of the screen. It's collagen, which is a very specific kind of protein. And collagen is used for a lot of things, particularly holding together tissues in animals. And that's the only thing that all animals always have that differentiates them from other kingdoms. So the presence of collagen is enough to definitively identify something as an animal. The rest of these features, you'll find that most animals usually have these things. Number 1 is heterotrophy. So being heterotrophic as opposed to phototrophic or photosynthetic or autotrophic. Heterotrophic means you have to ingest other organisms in order to get the organic carbon molecules such as sugar and other organic carbons that that animals need to construct the various biomolecules that they need to make. Multicellularity, so being multicellular is a feature of animals. They do not have cell walls. They have neurons and muscles for active movement. And there's a lot of diversity. So when you think about what's in the zoo are typically animals like lions and tigers and bears and giraffes, a rhinoceros, an elephant. But these are mostly, most of the animals you see at the zoo are mammals. And mammals only make up a very small portion of the animal kingdom, actually. Now, mammals do tend to be the top of the food chain, so to speak, the biggest predator, the biggest consumer in most ecosystems. And so they're very interesting, but they are not, in terms of diversity, not the biggest group of animals. And so, you have here on the right side of the screen a heartworm, and that's an animal. So actually there's quite a lot of worms in terms of the diversity of species, but mammals do tend to be the largest in size in terms of the organisms themselves, but they are not the most diverse group, not by far. So in any case, looking at some more features that most animals mostly have, there are animals in every habitat. So most animals are in the ocean, so marine refers to salt water. But you also have animals that live in freshwater and then animals that live in terrestrial environments. Most animals reproduce sexually, meaning they make eggs and sperm. Some animals, in addition, reproduce asexually. There may be a couple species that strictly reproduce asexually, but that's very rare. And I can't even remember the name of an example of one of those, but I do think there are some, but they're very rare. So the rule is most animals have sexual reproduction, and some additionally can do asexual reproduction of some type. And then tissues. Most animals have tissues. So tissues, you will recall, are defined as groups of cells that work together and we call that unit a tissue. So groups of cells of the same type working together, that would be a tissue. So not all animals have tissues, but most do. So the very early events of embryo development are shown here. On the far left, you have the zygote, which is what happens when the egg and sperm fuse and then their nuclei fuse. So the plasma membranes of the egg and the sperm fuse first, and then at some point after that, the two nuclei fuse together. And that creates the first cell of the new organism. And then the zygote goes through very quick mitotic divisions, cell cycle divisions, and basically they occur without much increase in size, so not much increase in volume of the cell between, like for example, G1 and G2, which are typically when the cell increases in actual size. So we call these types of very fast, very quick cell divisions cleavage. So it's a modification of atypical mitosis type division. And in this picture, it's actually pretty accurate in terms of size. So the zygote is shown at a reasonable size on the far left of this image. And what you can see, and what is actually correct, is that the entire embryo, even as the number of cells in the embryo increases, the overall size of the embryo does not get much more than what the original zygote was. Sorry about that. So the cells divide, but they don't increase in size. So you end up with, for example, the zygote divides a few times, and you get a 2-cell embryo, and then a 4-cell embryo, and then an 8-cell embryo. The 8-cell embryo is sometimes called the morula, M-O-R-U-L-A. And then those cells continue to divide, and eventually, somewhere around 32 or 64 cells, the cells start to move from the inner part of the ball of cells. They all migrate to the surface, and you end up with a hollow part on the inside of the embryo. And when that happens, that's called the blastula embryo. But the blastula embryo, the cells keep dividing. It's just that now the cells are all on the surface. And this is a cross section they've cut into this embryo, so you can see that the inside is supposed to be, you're supposed to see that there's sort of a hollowness. It's full of liquid there. And then the cells keep dividing. You start getting into the thousands of cells stage. And then as they keep dividing, then another event happens. Some of the cells in one particular point on the surface of the blastula start to migrate inward. And you can see on the cross section it's almost like an invisible finger is pushing from the outside, pushing the cells inward, just in one place. And we call that migration of cells to the inner part of the embryo. We call that gastrulation. And the embryo that has this little pocket of cells migrating inward is called the gastrula embryo. At that point, the gastrula is the point where the embryo is said to have 2 tissue layers. And we call those embryonic tissue layers, we call them germ layers. And don't confuse the word germ in this context with some other ways that the word germ is used. Sometimes we refer to gametes as germ cells, but that's a different use of this word. When we talk about germ layers, we're talking about the embryo, and we're talking about the layers of tissue starting from the gastrula stage onward. So the classification of animals, multicellular animals are collectively referred to as the metazoans. And you can see the term zoo, Z-O, which really means animals. Like if you go to the zoological gardens, Or if you study zoology, you're studying animals. So zoo means animals. It has that connotation or definition, zoo. So the metazoans, there's some clue in that term that it refers to the animals. And in this case, all the multicellular animals, which really all the animals are multicellular. And the multicellular animals are really, there are really about 36 phyla. And I haven't checked to see if they've added any new ones, but something around that. We are only going to delve into 9, what I would call the top 9 phyla. So it's going to seem like a lot, but it's actually, we've actually cut out quite a bit, but the top 9. And this would be, the same 9 that most textbooks, most courses will cover. So, you may go to a different university and they would cover 10. But 9 is a very solid number for the getting an idea of what the diversity is and how animals are different from each other. All right, so the metazoans, the first main division of the metazoans. So when animals first appear in the evolutionary tree, you have this one branch, which we call the metazoans. And then pretty quickly that branch splits into 2 groups. And one group are called the parazoans, and one group are called the eumetazoans. And the parazoans include specifically all the sponges. So phylum Porifera, Porifera, belong to this larger, or this clade we call the parazoans. But the parazoans is basically phylum Porifera. And so the sponges are sort of split off very early from the other animals. And it's because they don't have many of the characteristics that most other animals have. And 2 of the fundamental characteristics that we'll be discussing in this chapter have to do with body symmetry and the presence of true tissues, which I mentioned just a minute ago in terms of the gastrula embryo having the first 2 germ layers or first 2 tissue layers. So sponges don't have even those fundamental things. All the other animal groups are on the branch called the eumetazoans. And the eumetazoans have symmetry and they have true tissues. And then the eumetazoans branch into 2 groups. So that branch branches and splits into one branch that are called the diploblastic eumetazoans. And those only have 2 germ layers throughout their development. The triploblastic have 3 germ layers. So this is an overview, although I will be doing a tutorial where I'll make my own drawing. But it's going to be very similar to this. This is the one in your book. And I've put a star next to the 9 phyla. So what you can see on the left is the root of the animal kingdom, the metazoans. You can see it splits very early into the eumetazoans and the parazoans. And then the eumetazoans split again and again and again. So we will simplify this somewhat, but ultimately these are the 9 phyla that will be on the simplified tree that we're going to draw. And like I said, I'll draw it for you on a tutorial so you can practice it. And but these are the 9 that you're going to learn about, specifically in the next 2 chapters, chapter 28 and chapter 29. So this chapter we're looking at an overview of everything and some of the vocabulary really. But it's going to come, eventually we're going to come to this evolutionary tree. Now, on the left side, you'll see the ancestor, what we believe is the ancestor of all the metazoans are protists. So you may recall that the ancestor of all the plants were the algae. And so the ancestor of all the animals is a protist. Now, of course, the protists and the algae share an ancestor, but that goes very, very early into the history of the early Earth. So we're only going to track it back this far to the protist. And of course, some protists continued, but these we're tracking back to the protist that is the ancestor of all animals. So one genetic element that we like to bring in at this point is there's lots of genes that are important for embryo development, so don't get me wrong. But there is a particular group of genes called the Hox genes. Hox is short for the word homeobox, Homeobox. The homeobox genes, or the Hox genes, are present in most animals, not just mammals, but animals. And they are a gene family. That means there's a whole bunch of them. Each species has a whole bunch of these, and they're very similar between species. So, for example, mouse and human have very, very similar Hox genes. And the interesting thing about the Hox genes is it determines the layout of the body. So that's something we're going to see in the sense of when we look at the body structure of these different organisms, you're going to see the layout of the body is kind of an important feature. And so the Hox genes, the way they're expressed and where in the body they're expressed, because you have lots of different, slightly different versions of these genes in your body. And you can see in here that it's showing certain ones. You don't have to memorize what Hox genes are expressed where, but there's a concept that the Hox genes are genes that are expressed, and that determines. Sorry, my dogs are playing. Cut it out. And that determines, where the arms go, where the legs go, where the head is, and so on.